Okay, so I'm Danielle Reed, and I'm the Associate Director at the Monell Chemical Senses Center, and that's a not-for-profit research institute in Philadelphia. And I'm going to be talking to you today about how our inborn genetic variation influences our sense of taste. And I've called the title of my talk, The Genetics of Grocery Shopping. And you can see that this is actually the grocery store where I do shop. It's located in Philadelphia. And I'm going to be doing a few demonstrations with my helpers, Lauren and Jennifer, who in a moment will be tasting various things and giving you their perspective on the differences and similarity in the human sense of taste. Now, I want to give you a few tools so that you can better understand your sense of taste. So actually, there are two things going on, at least two things going on when we put something in our mouth. There's the sense of taste, which is something that comes from our tongue. You might experience something like sweetness, and then a sense of odor, which you sometimes get up through the back, what we call the back door of the nose, retronasal olfaction, which is different than stopping and smelling the roses, if you will. And one of the ways we study your sense of taste is through preference methods. So that would be you like one thing better than another. And I also wanted to introduce the idea of genetics and genotype. So genotype is an inborn characteristic, a little bit like blood groups, that defines your particular genetic material. So just to be a little bit more specific, your sense of taste arises from your tongue, and you can see where in blue that I have little pink bumps. This is my tongue, and those are called papilla, which is the Latin for nipple, and those contain sensory cells that send a signal from tongue to brain. So if you put your finger in a can of Coke, you're not going to experience sweetness, but if you put that same solution on your tongue, you get a unique characteristic and a unique sensation, and that is your sense of taste. So the same situation a bit with the nose. So chemicals come in through the nose, either the rear or the front, and that those signals are carried to the brain, where you interpret the chemical signals as a sense of smell. If you want to know where that happens, if you put your finger right here in the middle of your bridge of your nose, and you just could imagine traveling straight back, that's the roof of the nose, the nasal epithelium, where the taste cells are. Now, I mentioned that to study your sense of taste, we often use preference as a way to interrogate how you experience the world. And sometimes we do animal studies where a mouse might have two bottles of something and is choosing between them, and we might understand that they can taste something if they prefer it more or prefer it less. And we do much the same things when we study human subjects. So we might present subjects. This is my daughter in this particular case with several choices. So she sips each one and tells us, points to the one that she prefers or points to the one that has a more pronounced taste. Now, I also want to give you tools to understand. When I spoke about inborn genetic variation, I like to make sure you have the tools to understand this. So I'm going to introduce a couple of terms and terminologies. We're familiar with the fact that all of your genomic DNA, which is found in every cell in your body, in the nucleus of each cell, is a code that goes of nucleotides that are A, T, G, C in various combinations. But when people differ at a particular base pair location, here we see the highlighted in G, as a G and a C here. You can see that one person might be a G at this location, whereas another person is a C. And we refer to that, generally speaking, as an allele. It's an alternative form of this particular location. Now, it turns out that people are quite different in their genomes, and so you might have a difference here, and then you might have a neighboring change right here. And so, for instance, this chromosome is GC, whereas this chromosome is GT. And when we're talking about this combination, we refer to this as a haplotype. Now, in some instances, these genetic changes are in the protein coding parts of our DNA. So about 5% of our genomic material is involved in protein coding genes. And when this occurs, this triplet code, when we go from G to C, it actually specifies a different amino acid. So for instance, GCA codes for alanine, whereas CCA codes for proline here, the A standing for alanine, the P for proline. And likewise, you can see that we might talk about a haplotype as being either a nucleotide here or an amino acid here. So this person would have an A haplotype. And because everybody has two autosomes, one from mother and one from father, so one from mother and one from father, you have this combination. So for instance, at a given location, a person might be AP, and this combination of two haplotypes we call a diplotype, di meaning two. So for instance, now that we have this knowledge, if we can envision these as our chromosomes, that this person would be alanine, valine, isoleucine on one chromosome here, and the second chromosome is identical. Somebody is heterozygote, and then another person could be homozygous for this form, the PAV form. So these are inborn genetic variation. And this inborn genetic variation is in many of the genes that code for our taste receptors and other molecules important to transduce our sense of taste. So just to give you an idea about the breadth of human experience in our sense of taste and smell, we're going to take a little trip through our grocery store, smelling things and comparing our individual differences and looking for the underlying molecular explanations of these. So as we're walking into our grocery store here, the first thing that you come to in my grocery store is actually the meat section, and here we have some bacon. And bacon contains something which is called boar taint. And for instance, if male pigs are not castrated, their flesh can contain an unpleasant odor, which is the combination of two chemicals. One is called skatol and one is called androstenone. Now, some of you may be reflecting on your experience and not sort of wondering what I'm talking about, because to you, bacon is absolutely lovely at all times and in all situations. But as you'll see here, I have a vial of androstenone, and I'm going to smell it, and then I'm going to actually ask my two demonstrators to smell this too. So I'm going to smell, and I'll report to you what I can experience, and then they'll do the same. So when I smell this, I find it extremely revolting. I find it to be urinous and deeply unpleasant. So let's have Lauren and Jennifer smell and report what they experience.
Okay. I smell nothing. Absolutely nothing.
I also smell nothing. Nothing at all.
So you'll see from the demonstration, I'm a very sensitive smeller of androstenone, whereas Lauren and Jennifer are not. And so what's the explanation for this? And the explanation lies in our inborn variation in one of our olfactory receptors. Now, there's an olfactory receptor, and there are 400 olfactory receptors, and they all have names, a little bit like zip codes. And so this is one particular olfactory receptor, OR, for olfactory receptor, and this is its little zip code name, 7D4. And some people have a particular genotype, which is arginine-arginine, or RR, and some people are heterozygous, and some people are homozygous for the alternate form. And you can see here that if we just look at the proportion of people that can smell this odorant, this androstenone that I just smelled, you can see that, that cannot smell the odorant. People that have this alternative form, this WW, are unable or mostly unable to smell this particular compound. So we might imagine that without peeking at the genomic DNA of Lauren and Jennifer, that they might be WWs, or at a minimum heterozygotes, whereas I am more likely to be the sensitive form, the RR form. And I've just shown you here this androstenone. I've also just shown you another compound here to show that this is a specific inability due to a specific inborn variant, so that this variant is not related to another compound, which is galaxolid, which smells pleasant to some and not to others. So this is an example of an inborn genetic difference that accounts for big differences in the perception of our ability to smell a particular compound. Now, it's also very fun to go into the aisle where the food and drink for the drinks are, and this is something that you might know as tonic water. And tonic water contains quinine, which is a bitter thing. And you might wonder how it came to be that people would like to drink something that's bitter. And as it turns out that soldiers that were serving in South Africa we're dealing with malaria, and quinine was used to be a first line of defense against malaria. And so soldiers would put a little quinine in with gin and a little bit of water and make a nice gin and tonic drink. And so they were able to stomach their malaria medicine, and people grew to sort of like that combination. So although quinine is very bitter, it is not, it's bitter, it became to be preferred. Now I'm just going to take a break here. So the first one, is that quinine? Okay. So I can tell you a funny story. So we used to use quinine as a sort of standard to see if people could taste, because our belief was it was like a control substance that everybody could taste. But it was a very funny thing. So one day somebody walked into my office who was helping me, and she was very sorry that she'd made the solutions wrong, and she was sad because when she was tasting it, it only tasted like water to her. So this was very unwelcome news. So I was suspicious, so I took it and I tasted it, and for me it is insanely bitter. It's extremely bitter. But for her, it was very, not hardly bitter at all. So I think we'll just go to Lauren and Jennifer and see if they have any particular response to this.
You go first. It's pretty bitter.
It's mildly bitter.
Yeah.
It's not very bad.
Mm-hmm. So you see here that this is sort of a shades of gray. So I experience it as quite bitter, and they seem a little bit less emotive than I do about it. So you can see that in the last example was either or, they could smell it or they couldn't smell it. And here it's more of a shades of gray. So one of the ways that we try to figure out what's going on, so there was no prior knowledge about any inborn genetic variation. And so what we did was something called a genome-wide association study. And what you need to know about that is that we look at genotype all throughout the human chromosomes. And then we compare individuals who are similar in their phenotype, in this case people who are similar in their intensity that they experience quinine with their particular genotype. And places where you see these big high bumps, this is called a Manhattan plot, are regions of the human genome where genes and their variants are responsible probably for this particular trait. And for quinine, you can see we have this particular taste receptor. So taste receptors, like olfactory receptors, have little zip code names. This is TAS for taste. And 2, this is the second family. The first family is for sweet, and the second family is for bitter. R for receptor. And then there's a numbering, so a system, because there are 25 receptors, and this happens to be number 19. And you can see that people who are AA compared to people that are GG have a differential perception of quinine intensity. So now we're moving in the grocery store aisle to the sweet solutions. And as I mentioned earlier, the bitter family is TAS2R, is the TAS2Rs, and I mentioned that there's a TAS1R family. And this is a receptor that's a dimer, which means 2 proteins intertwined together to create this receptor. So you can see this is the cell membrane here. This is what's called a transmembrane domain, which anchors it sort of like a sea monster through the plasma membrane. This is the part that hangs down in the cell, and this is the part that actually, if you put something in your mouth that's a chemical, for instance, a molecule of sweetness, this actually touches this part of the protein and starts the signaling, so that when you put the sugar in the mouth, you get a sweet signal, whereas putting your finger in the sugar water doesn't do the same thing. So this TAS1R3 we discovered originally because we were examining strains of mice, inbred strains of mice. So just like there's many inbred strains of dogs, like Chihuahuas and German Shepherds and so forth, there's comparable inbred strains of mice. And some are very enthusiastic consumers of a sweet substitute called saccharin, where others are not. And when we looked at the preferences of the mice for different levels of saccharin, we saw that the region that they shared of their genomes was this particular place, and in this particular place was a gene that we named TAS1R3. And so you can see that many strains of mice have one particular variant of the sweet receptor that makes them very enthusiastic about saccharin, whereas other strains of mice are much more ambivalent. So this is the case in mice, but we might wonder whether humans might have inborn variation of these sweet receptors that might make people different in how they perceive sweet taste or liking of sweet taste. And so here, this is sucrose preference. You'll remember I showed you a slide earlier of a little girl choosing between different cups of sweetness, and so you can imagine many people doing that. And here we're looking at the particular inborn genotype at this TAS1R3. Remember, that's one of the partners that makes the sweet receptor. And a lot of variant sites have these very complicated names that start RS something, 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 something. And there are millions of those, and this is just the particular sort of zip code for this particular variation. And so people are born with different versions of the sweet receptor genotype, and you can see that this has to do with their sweet preference. So this here, this CC here, these people prefer sweetness at a very low level relatively, whereas people that have this particular genotype prefer almost 3 times the concentration of sugar. So this is about the idea, this is about the sweetness of a common Coca-Cola or other soda sweetened beverage that you might buy. And so I thought it'd be fun to just see which of these 2 levels of sweetness I prefer. So I have here a low and a high level of sweetness. So this is the low level. That's much better. So I have to say, I have to confess, I really prefer the sweeter solution. And so we'll see if this is a universal opinion. So we'll have Lauren and Jennifer taste this. So now they're tasting the first sweet solution.
Yeah, I prefer the sweeter.
I do as well, actually.
So we have a lab full of TT genotypes in our lab. So you can imagine that when you're trying to, from a practical level, if you're trying to cook something or sweeten something in your home, you're appealing to different palates, and it's not just that people are being irascible or preferring one thing over another willy-nilly, but there may be actual genetic variation that predicts how much people that you're cooking for or making things for or manufacturing products for might perceive and might like in their diets. Now, I want to talk a little bit more seriously about the idea of bitterness. And bitterness often comes up in 2 places, medicines and vegetables. And the reasons that many vegetables are bitter are that plants really cannot defend themselves very well. You can't run away if you're a plant, but what you can do is manufacture something that makes you unpalatable to predators. And these are so-called plant secondary compounds. And many of the things that make vegetables very healthy for us at very low doses are at higher doses are very bitter. And we're always trying to encourage children to eat more vegetables and adults to eat more vegetables, but we're often fighting this idea of bitterness. But as you might imagine from our demonstration of quinine in tonic water, not everybody perceives the bitterness of vegetables in the same way. So this is an experiment that was extremely unpopular, where I had people chew up 40 samples of raw vegetables and spit them out and rate them for bitterness. So on the y-axis, people were rating each vegetable for mean bitterness. So big numbers mean things were more bitter. And this x-axis is a little bit trickier to explain, but it's basically the spread of people. So that small numbers for variation means everybody agreed and that agreed with each other very well. And big spreads means that there's a great diversity of viewpoint. And what you'll notice is there's a reason probably that we are often offered carrots as a popular vegetable. It's because they're not very bitter. And everybody agrees that they're not very bitter, so it's a safe choice. So when you're trying to make something to appeal to everybody, go for carrots. But you can also see that there are some vegetables that are very polarizing. So for instance, horseradish and watercress and turnip and collard greens. And I find that vegetables that are in this category are often unfamiliar to people because they're often very rarely served, or they're served only in some households but not others. So you can imagine if you were going to be making a salad, you probably want to avoid the watercress if you're having everybody over, and stick with your carrots and your potatoes. Now, why is kale out here in no man's land? I don't know. So kale is very popular, and it seems to be a bit of an outlier, but you can see the general trend here. Now, actually, all of the individual variation in taste and smell traits arises from the knowledge that this was probably the case actually predated the discovery of these receptors. And what happened is in the 1930s, there were chemists that were working in the DuPont chemical plant, which is in Wilmington, Delaware, and they were synthesizing a new compound. And one of the chemists was transferring the powder from one little jar to another, and did one of these, like flour, you might imagine transferring flour, and the chemical puffed into the air. And his benchmate, who he wasn't necessarily getting along very well with to start with for other reasons, really objected to the bitter. And as you can imagine, the first person couldn't taste it at all. And they were very surprised at that. And they reflected on why that was, and they came up with the idea that was later shown to be true, that there's a host of different receptors. So there's, in fact, 25 bitter receptors, and one of which happened to be broken. And so there was a selective inability to taste this bitter compound. And this bitter compound is called PTC, or phenolphthaleocarbamide, and has this structure here. Now, you'll remember that I talked to you about different genetic variation. So here's a tree showing all of the bitter receptors. Remember, TAS2Rs are bitter receptors. And just like languages, the bitter receptors that are the most similar in structure are the most close on this particular phylogenetic tree. And you'll notice there's a bit of craziness here in the numbering scheme. So I told you there are 25 receptors, but some of them are labeled up to 38. So why is that? And the reason for that is when they were originally discovered, there was sort of a naming frenzy, and people were naming things and rediscovering them and naming them again. And so when the dust settled, we had a bit of a crazy numbering system that we still retain to this day. So this is one of the bitter receptors, TAS2R38, and it has this characteristic structure, this 7 transmembrane domain, outside the cell, inside the cell. And some people are born with an A, V, I form, whereas other people are born with a P, A, V form. So AVI, and remember there are 2 chromosomes, one from mother, one from father. So this person is homozygous for the AVI form, whereas this person is homozygous for the PAV form. And here you can see that this inborn difference strongly predicts the ability to taste this bitter compound called PTC. So as the molar concentration of this bitter compound goes up, people's perceived intensity goes up. So we're just going to take a little micro break, and we're going to now sample the PTC paper. So this is often done in classroom demonstrations. So the other solutions I showed you were solutions, but we have this little convenient piece of paper here. Now, you will tell from my expression that I am a non-taster. To me, it just tastes a little bit like paper. So we're going to now turn to Lauren and to Jennifer and see what they have to say about this. I actually don't know what they are.
It's really bad. It's very bitter. Very bad.
Very bad.
It's getting worse.
Very bad and getting worse. So the point here is this is a very marked difference in people's perceptual ability. Whereas quinine was sort of shades of gray, this is a very, very marked ability to taste this particular compound. And while PTC is a compound that was made by humans in a chemistry lab more than 70 years ago and doesn't occur in anything people might actually put in their mouths, it's actually structurally related to a compound called goitrin, and goitrin is a very common secondary compound in broccoli. So again, you can see here that people, when they're rating, they're able to, this is a threshold measure, the lowest concentration at which the people can detect goitrin. So you can imagine a little tiny bit of goitrin in a big, in a solution. People that have this genotype are very, very sensitive and can perceive this broccoli compound at very low concentrations. And so one of the things you might imagine is that based on your inborn genotype, you might have a very different opinion about a variety of vegetables that contain this bitter goitrin compound. So here we've listed all sorts of, in one of these mean studies where people are constantly asking you to chew up vegetables and spit them out. You can imagine here we have watercress and collards and so forth, and these are all vegetables that contain this goitrin compound. And you can see that those who are PAVs, people like Jennifer and Lauren, are very different than people like me, the AVIs and AVIs of the world, so that I'm not experiencing as much bitter from these compounds that are in vegetables as they are. And so you can imagine that there could be a real difference of opinion about vegetables that has nothing to do with how you were raised or culturally or so forth, but has everything to do with the genotype that you were born with. Now, we really don't know how your inborn genotype relates to your real-world decisions, because many things influences your choice of foods, cost, what you're familiar with, and so forth. So one of the big gaps and one of the opportunities for students who are interested in this particular idea would be to look to see how people actually behave in their real environment. So with that, I'm going to wrap up just to say that my research institute is called the Monell Chemical Senses Center, and we're very encouraging of students from all places and all different levels, and we have internships available in the summer and for high school students and for college and for undergraduates, and sometimes teachers come too, so we'd love to see you. So thanks very much.